I wanted to share a bit with you about what is developing at the Institute for Sustainable Horticulture, because you've heard where our inspiration comes from. So we are developing beneficial fungi for use in pest management. In 2011, we started with a survey of soil and insects to extract the beneficial fungi from the soil. And also, we went and did collections of insects for fungal illnesses. And very ambitiously, we took 575 soil samples from various agricultural sites and forest sites and parks and turf and golf courses. And there's a story there that we haven't quite figured out yet about where we were finding different things, but we're still identifying certain things. But it's going to be an interesting story, because out of it we have many fungi. We have a group of them that are trichodermas, and we have a group that kill insects. So we have Metarhizium, we have Bovaria, Lecanicilium, and Paecilomyces at least. And then we have about 99 isolates of trichoderma. We're doing murder trials with the entomopathogens to see who they kill, because they may kill nobody. I mean, not all Metarhizium are toxic to insects. Some of them are just happy to live in the soil. They've got some other interest, no interest in insects whatsoever. So we're screening them against insects, and we're using Galeria, the one that you're taking home after you've infected, because it's a very wimpy insect and it'll usually get infected by anything that you put in a dish with it. And then all summer we brought in insects from different crops and Teresa did this. She was the murder trial. She was the judge all summer, setting up these murder trials, and we're building a table of our isolates and what they're killing. And that'll give us an idea where they might be useful as products. So we've had 2 field trials already outside, one with Metarhizium that we extracted from European chafer, actually before 2011, but it was in field trials in 2011. It's affectionately known as MU1, M001, and it's reasonably good in the field on European chafer. We did something different with endophytic grass this year, and we got no results, but the endophytic grass seems to be pretty toxic to them. Anyway, with the trichoderma, we have close to 100 isolates. Probably 40 are going to be useful to us in developing new products. We've been screening them against the 3 main types of pathogenic fungi. The oomycetes are the ones like Phytophthora and Pythium, and then actinomycetes and basidiomycetes are other classes. And so we're now building a picture of which of those strains, which of those isolates are effective in a Petri dish, so not in the field yet, but in a Petri dish, against different types of plant pathogenic organisms. We've had 1 field trial. So we're continuing, we're just about finished the lab screening that we can do until we get some more pathogens. And then we're going to decide where we're going to go next. We have the field trial that we did in 2012 in potatoes was part of a collaboration with Cuba, and you heard Jorge talk about now studying trichoderma for use as a biofungicide in Cuba to reduce the fungicide load on potato. Well, in Canada too, our biggest pesticide load on potatoes is fungicides. The insects we can control with next to nothing, less than 10% of what growers used to use with integrated pest management. For the diseases, no. And in this part of the world, our major disease is Phytophthora infestans. It's late blight of potato, caused the Irish potato famine every 8 years or 10 years. That fungus mutates into something worse. and so it's quite a difficult one. And growers tend to have to apply every week a fungicide to protect the new growth. If you're an organic grower, you're applying copper. And if you know anything about copper in plants, you know it stunts the growth of plants. It builds up in the soil. It eventually affects your yield. So while it's allowed in organic production, it's not ideal. So there have been studies here for a few years to try and find alternatives to copper that will be acceptable to the organic industry. So we tested Phytophthora infestans in Petri dishes against some of our isolates, and we had to pick 2 winners. They were 207 and 503. 503 we believe is Harzianum, and 207 we believe is Ovalosporum, but we're not totally committed to those names right now. And we got permits to use them in field trials this summer. Molecular work is in progress, and rather than do molecular work on 100 isolates, we're narrowing it down to the ones of most interest, because that gets expensive too. So we had Alexander Calero, a Cuban graduate student, as part of our Students for Development project, spent 4 months with us this year, and he was intimately involved with these field trials. In Cuba, his trials are on alternaria, so early blight of potatoes, but because that's the major disease in Sante Spiritus, but here it's Phytophthora infestans. So he came here and he was right in the middle of the field trials. So the first thing we wanted to know is which of these isolates grew very quickly, because if they grow quickly, they've got maybe more of a chance in the field. These are just indicators of success in the field. 207 is at the top there. It grew very, very quickly in a Petri dish. You might notice that 207 didn't grow quite as quickly in the bag there. The one in the middle is actually the Trichoderma. It's the Cuban isolate, which we did not put in the field. We're not planning to put in the field, and it's only in here for comparative purposes. And the one at the bottom, 503, was the slowest growing one, but you notice in the bag it's the one with all the nice green mycelia. It actually was quite fast in the bag. And then we did some dual culture assays, and we grew Phytophthora infestan in Petri dishes and let it grow for, I don't know, quite a while. It's a very slow grower. And then we put the trichoderma on each side and let it grow towards. And the white is the mycelial growth, and how quickly that grows towards the disease, it stops the Phytophthora infestan from growing, and we can measure that. And then we've got some other measures as well. Sometimes it attacks it, it eats it, it kills it. So, but this is what happens when you put it in the presence of the trichoderma. The control there, the top line, that Phytophthora infestans keeps growing, the circle just keeps getting bigger, it grows out from the circle. But as soon as the trichoderma, both of those strains hit it, it just stops it altogether. So the growth stops. The trichoderma is obviously quite aggressive. So in the field, we had two varieties of potato, one Norcota, which is a very popular brand, a popular variety, but it's a wimpy plant, and it's also quite susceptible to late blight. It doesn't have very many leaves, it has a short growing season, almost anything, it will knock it over, so it's quite a weak plant, but a lovely potato. Innovator is a really ugly potato, but it seems to have some innate resistance against Phytophthora infestan. So we had a control untreated. We had a copper control, so treated with copper. We treated every week with Trichoderma 207 and a Trichoderma 503, and then we alternated copper with each of those, because maybe if they're not strong enough to control the disease, maybe alternating the copper will at least reduce the copper by half that's used. So we grew our Trichoderma only in liquid culture. We had it in the either in the flasks or not, and then poured it into those trays and then let it sporulate on the surface. Took that, put it in the blender, blended it all up, counted the spores, because that was really the only indicator of activity that we could count. And then we went and sprayed it in the field at a rate that has been used in field trials in Cuba. And that's Alexander. Those are our plots on the farm. It's a farm that has late blight every year. It's absolutely reliable in that regard. And we, of course, got late blight in our plots without having to inoculate. And so we could count things. A few things were measured and that were pretty interesting. The control and the control 2 are untreated control and control treated with copper. And in the middle, you've got 207 and 503 alone, not the alternating ones, just those two alone. And in our plots, we measured the number of leaves per plant just to see what effect there might be on the plant of having those sprays, because not only do we have spores in those sprays, we have metabolite, we have mycelia. We just took everything and blended it. So in fact, both of them increase the number of leaves on the plants and increase the number of stems per plant. So when you do that, you're increasing the food that's going down to the tubers, and there was a very similar increase in yield in those two treatments that we were treating with the fungus, just kind of as an aside. And it wasn't really related to the disease. It didn't seem to be related to the disease, because the disease came in sort of late, and we were counting lesions, and the plants, I mean, there was yield even under the diseased ones. So in terms of efficacy, how well did those products control late blight? It'll be hard to read the bottom, but the blue one is the copper control, and that 70% is 70% reduction in lesions, so countable lesions on the plant from the untreated control. So copper controlled it at about the 70% level. The two yellow ones are Trichoderma 207, 207 alone on the left and 207 alternating with copper on the right. Now these are not significantly different, any of these numbers, but consistently those green ones, 503, any treatment with 503 was slightly better than the treatment with 207, and they're completely comparable to copper. So you could choose copper or you could choose Trichoderma, at least in this year when the pressure seemed quite high in the beginning because it rained so much, but we actually didn't get the disease in the plots until later. It was quite a surprising year. So our conclusions are that 503 performed better than 207. 503 alternated with copper or alone, both provided comparable control of late blight to copper. And Innovator, actually those plots, they didn't get, they were probably 4 weeks later getting their first lesions than the Norcota. They were just 0, 0, 0 at every assessment, and then all of a sudden near the end, just when we were going to take the plots out, they got it. And this has happened like 3 years in a row in these trials, so there's some innate resistance that you wouldn't want to eat, you wouldn't want to buy Innovator, but there's some genes in there that might be useful for breeding resistance. So one further thing we did is to look at the effect of the metabolite on roots, because there's something going on when we sprayed those plants that isn't necessarily related to the spores. We didn't think. We thought it might be related to the metabolite. In Cuba, they do a lot of work with the metabolite and as a biofertilizer, as a stimulant to the plant. So Alexander did this work, and we've got a nice little root bioassay. I'm not going to tell you too much about it, but here we have treatment 1 is a water control. Mung beans will grow roots in water. So they're germinated, you cut them off, you put them in water, and they grow more roots. So you can quantify the number of roots that they grow, the number of laterals, different things, the dry weight. So T1 is water, so the mung bean plant grew some roots in water. T2 is a positive control, one that we know quite well. It's kelp grow. It's a kelp product, and we're doing a lot of work with it in the lab, and so we know that that's a positive control. T3 and T4, I'm going to go back here. 3 and 4 are 207, and we did metabolite at 1%, 5%, and 10% of that water solution. So 3, 4, and 5, 1, 5, and 10%. 10% is bad. It actually prevented root growth. But 1 and 2 aren't, 1% and 5%, not too bad. Not as good as the kelp grow. And then 6, 7, and 8 are 503 in 1, 5, and 10%. And you sure don't want it at 10% or even 5, but 1% looks reasonable. 9 and 10 are combinations of 207 and 503. 9 is at 1% and 10 is at 5%. And look at the roots. When you add them together, you get a lot more roots. And it was absolutely reflected in the dry weight. The best treatment by far was a 5% of 207 plus 503, those two isolates mixed together at 5%. So that's a total of 5%. So what does it mean? Not really sure, but 207 and 503 both had more leaves, they had more yield and more shoots. And when together, they've got more roots. Not so much alone, but together. So this is something that we want to follow up with and just see where that'll take us. We're working now on temperature for development of approximately those 40 isolates of Trichoderma. 503 and 207 are going into another trial this winter to control soil pathogens. And we had a nice little preliminary study that showed that we got pretty dramatic control of some damping off diseases. We'd like to put in a pre-registration consultation request for 503, just to be very, very bold and brash, and hopefully repeat the trials in 2013. And we didn't combine 503 and 207 in the field this year, but I think we should do that. And I presented this data this week at a potato industry meeting, and that was the idea, that we should definitely take this forward into a proposal for the next few years. So if you have any questions, we can answer. If you have any questions for the other speakers, maybe I'll get them to come up in case there's some questions. Yeah, Diego?
Yeah, what do you mean metabolites? What do you mean exactly?
What do we mean by the metabolites? Well, when we grew that trichoderma, we had molasses and torula yeast and water. And we put some spores in. So as the spores germinate and the mycelia grow, that growth is accompanied, they use up the nutrients in that solution and they produce something.
It's kind of byproduct.
It's a byproduct. We just call it metabolite because there's something in there that we didn't put in there at the beginning. And when they've used up the nutrients in that solution, that's when they'll sporulate on the surface. So we knew that pretty much the yeast and the sugars were gone, and whatever was left in that liquid was mycelia and metabolite. So we don't know, and it has not been characterized. It could be characterized.